Okay, this is Speed of Sound, and I'm with Daniel Stern Cardinale. Is that how you pronounce it?
You
Cardinale?
got it. Stern Cardinale. Yeah.
Dr.
You just call me Dan.
Dr. Dan. How about Dr. Dan?
That works too. Either way.
Okay. Anyway, and we're going to have a conversation about genetics and biology and evolution and the strange world of the internet. And we did not have the talk prior about production, but maybe we can do that after we hang up here.
Yeah, so. Absolutely. Happy to.
Okay. So one thing I did a real long analysis of cells' fitness ideas and all the various fitness ideas in population genetics, and I found that the Fisher's fundamental theorem is not so fundamental after all. But that's not the point. I mean, it's a nice little theorem, and it makes sense, and it's statistics, and it applies where it applies, and I don't see any other problems with it. So,
right, and so if you want to get into the Stanford paper, I'm happy to talk about that paper because there was one giant error with that. What is it? Basner and Stanford 2018.
Right.
There is one giant glaring error with that paper. And it just makes me shrug at the whole thing. And the problem is that they're basing their math on two distributions. And actually, I can share this. Let me just pull it up real fast here. I will share this to show the appropriate figure because it really gets at the whole thing. There it is. It's this one. Here we go. So let me share this if you don't mind. We're going to. Yeah, we're going to pick the this one. So what we're going to do here, actually, you know what? We're not going to do that. We're going to do it so I can, sorry. 1 second. Browser, oh, come on. Yes, share screen, please. Your browser has blocked. Oh, I have to reload? No, why do I have to reload? Oh, what the heck with it? I'm going to share the screen. So figure, it's not letting me, and I don't know why. I'd have to reload and rejoin.
Trying to find the paper and yeah, you could do it real fast.
Yeah, it's figure. I'll do it real fast. It's figure. Okay. It's figure 1 in Basner and Stanford. That is the
Basner and Stanford. Waiting. Oh, there you are.
Okay, I'm bad at this. I'm bad at this.
I am too. My production values are crap.
For literally 2 weeks. I'm terrible at this. Let's see. Here's what we're going. We're just going to share the entire screen. Is what we're going to share. Yeah, we're going to do it like that. Okay, so now you should be able to see me and you and Basner and Stanford over here.
Here we go.
So what we have right here. Oh, that's a.
Oh, you got the.
Sorry, I see. Here you go. There you go.
Oh no, we fell into the infinite well.
So here's what we got here. For figure 1, Basner and Stanford, we have 2 distributions. We got a Gaussian distribution in light purple, which is basically symmetrical, and it's unrealistic in that regard because like mutation effects are not symmetrical. But then we have a gamma distribution right here, which is obviously very asymmetrical. Most mutations have a very small harmful effect, right? They're all on this side of the distribution. Now let's see if I can find where, so that here we go. This is from Kimura, 1979. Now I'm going to close this. I'm going to stop the screen share because it's going to go down a rabbit hole. There we go. Now the problem with that is the same distribution that Stanford uses in the book Genetic Entropy and bases his whole thesis on. That distribution is not based on data. That distribution is based on the parameters for Kimura's model from 1979, where he deliberately excluded beneficials.
It was theoretical. Yeah.
Yeah, exactly. So now Basner and Stanford come along with this paper. They show if you have the symmetrical distribution, then fitness in a population increases. If you have the gamma distribution that's based on the 1979 model, fitness does not increase. The problem is what they're selling is that second half.
Right.
The fitness does not increase second half. The parameters underlying their model to get there are themselves the parameters for a model that deliberately excluded beneficial mutations. They're not based on data. They're not empirical. They're not realistic.
So they did it again.
They did. It's literally the same trick that Stanford does in the book. They did in the 2018 paper. It's not different.
Yeah, they covered under a lot more smoke and mirrors, though, in that paper.
Yeah. Oh, yeah. There's a whole bunch of math, and it's complicated, and like it's legit. But the underlying, it's a case of garbage in, garbage out.
Yeah.
If the underlying parameters for your model do not reflect reality, then you're not going to get a realistic answer.
Right.
And that's it. And that's all it comes down to in that paper. You just look at the parameters for the model that they started with. They hit that first domino, and they got the result of fitness cannot increase.
And that was peer reviewed, right? In a real journal.
And what journal did they use? Because Stanford has a habit, and they did this with the H1N1 paper too. They have a habit of picking journals where the reviewers are not going to be experts. So this is the Journal of Mathematical Biology. It's not an evolution journal. It's a math journal. So what the reviewers are doing there is checking the math.
Checking the math, right.
They're not checking that it's realistic.
Yeah. I wonder if their program is open source, if it's downloadable, because somebody suggested that we look into the program too. I think part of it, they said it was written in JavaScript. I thought it was written in, I know part of it was written in, I think Go or something, but anyway.
I don't know off the top of my head. It would be cool to play around with it though.
Yeah.
It would be cool for someone who knows what they're doing to play around with it. And that someone is not me.
I would like to look at it because I happen to be a programmer. So, kind of what I do. So
definitely look at that.
Yep. But yeah, that was I suspected that was the case, but then I got scared off by the math. And
there was nothing to it. There was I honestly, like, you can go through the math, but the math doesn't matter. It's that baseline assumption of the distribution you put in is the only thing that matters and is wrong. It's just not realistic. That's it.
The thing that concerns me about creationists is that you listen to them. Like if you listen to Standing for Truth's channel, you'll get this kind of like this peck. And I started calling it chicken picking because they like peck, the corn. They find a little kernel of corn, and they'll take a paper and they grab a piece out of the paper, like on this Felsenstein paper, he grabbed like one quote, and then that quote becomes their argument against evolution, and they mount like 20 of them. And they say, "Oh, we obviously we've proven our case," and it takes me for every peck, it takes me 4 or 5 days of research.
Oh yeah.
Oh, what the hell?
Everyone is a rabbit hole.
Yeah. And then when you like chromosome 2 which is absolutely amazing. Like the DDX11L2, right? So I looked at the DDX11L2.
Is that the one? Is that the one that spans the fusion site?
Right. Yeah, but does it really? It's. I went to NCBI. I downloaded it. I wrote a Python program, decoded some of the. I basically made colors for each amino acid. And then I went over and I did the same with DDX11. And I'm like, whoa, DDX11 is this long. DDX11L2 is one motif. One little, I don't even think it has a valid domain in that entire protein when it's transcribed. I think it's like 200 amino acids long. And the actual gene is like 1,000 or 900. And it has 9 or 10, I think it has 9 independent domains, including the dead box, the DEA, or DEXH. So there are all these domains that are important for that to be a functional helicase, right? So this little snippet, and then to make matters worse, there's it's transcribed in the reverse direction. There's exon 1, which is on one side of the fusion site, and then there's the fusion site. And that exon 1 is very seldom transcribed in any tissues. So it just gets a little hit now and then where it'll transcribe, but I think it's like 14 amino acids. And then it goes a long ways over the fusion site. And then you got the other 3 exons, which are usually transcribed. But even those 3 do not put together anything that looks like a viable enzyme or protein, and they keep, every time they refer to it, they say the highly functional DDXL11 RNA helicase.
Yeah, it's not actually functional.
And I almost go through the roof every time they say highly functional. It's expressed in over 200 tissues. Well, mostly it's expressed in the testicles. Do you know why that is? CRISPR told me that the testicles are kind of like the slut of the body, where they'll sleep with any gene. Basically, any gene will get transcribed in the testicles.
Yeah, a lot. It depends on which specific tissue, but a lot of what you're doing is resetting like methylation patterns. So throughout the rest of the body, you've got lots of gene inactivation, but in like the seminiferous tubules, you reset those patterns. So you get a lot of spurious transcription that you don't have in the rest of the body. It's not that you're making the enzymes, necessarily. In some cases you are, but in a lot of cases you're not. It's just transcribing because you're remodeling the methylation patterns.
Right.
And while you're doing that, DNA is exposed that normally isn't, so you get a lot more transcription.
Yeah, that's where it's expressed the most. And other than that, it doesn't have any significance at all. So then I went over and I looked at the centromere. I finally found the centromere, and I forget whose paper it was, and he located the vestibular inactive centromere, and you find that anchor 30BL gene over there. Same thing. Same exact thing.
Is that the ancestral centromere from one of the
from one of the chromosomes, right? And so it was exactly the same thing. There was one little exon on one side of where the centromere was. The centromere is really wrecked. I managed to create a Python program, and if I scrolled at 190, like if I changed the width to 190, you could kind of see a diagonal pattern going on. So it was hard to pick up, but I forget the author, but he did manage to pick up signals on that centromere. And then I looked at that, and it was the same exact thing. It had one exon on one side, and then it had the most commonly transcribed or spliced exons on the other side, and it was not a functional gene. It was tiny compared to the anchor, the actual one, the one that isn't a pseudogene. So it was a chopped off gene, and then both of those genes are present in the subtelomeric region of the chimpanzee. So it was just like,
they're there, it's just that whole argument. And then who's the guy that tried to do this a couple of years ago and got smacked down by everybody, including Tompkins, who he ends up getting smacked down by some creationists too that basically said stop making bad arguments.
Stop it.
It's the chromosome 2 thing is exactly the slam dunk that like evolution, okay, can we make a prediction? Yes. What do we find? And it's exactly where we expect it to be. And oh, there we go. And you can try to like rationalize your way around that, but it works. It looks like it should look. And transcription around the telomere doesn't get around that.
Yeah. This is, let's see if I can, I wonder if it's all there. I think it is. Okay. So what you're seeing here is I got the DDX11 out of the rat right here. And that is only 50% identical, I think, to the human and the chimpanzee. And, or wait a minute. Okay, this is the rat. I'm not sure what that is. I don't know what I did. Okay, this is the rat. This is the human. And I think this is chimpanzee. But this is DDX11, right? L2 right here.
Yeah, that's that teensy little fragment.
That's all it is. All of the motifs are over here. There's, I think the dead box motif is over here. And it's, I mean, that's it. That's a highly functional gene. And the exon site is like, or the telomere, the fusion telomere is like right here somewhere. I don't know. Yeah, I think you can see my cursor.
Yep,
it's right
I see
there.
it.
Yep. And let's see. Then I went ahead and I did some comparisons. Rat, chimp, human.
Oh, that sequence is a mess.
Yeah.
You can look at that. That's all. That's not okay.
Yeah. Well, you can see that there are similarities here, but it's highly degraded, so it's not conserved.
Which is exactly what you would expect in a non-functional fragment.
Right. It's similar here, but I mean the rest of it it's a mess over here. I had a heck of a time aligning it. I am taking courses in bioinformatics, and I had not yet gotten to the part where you can use alignment software when I did this. So I did it.
You got to do it manually. There's no way around it.
Yeah, that's the other thing. So here's the whole protein. That's it. I mean, there's nothing to it, and I think that's with. I think that is without that first exon. I'm not sure. And here's the real protein. Basically, it shows. I think it shows the dead box gene. I thought I had it the most. The most conserved site is highlighted in green. So that is the one that's present.
That's not the same thing as the fragment by the telomere.
Yeah, look how long that is. So then I went down, and I don't know what the heck this is.
No enzymes like that are going to be so short anyway. The only things that are functional or that short are little messengers.
Right.
Enzymes are not going to be that short.
Yeah, and so let's see up here. I'm trying to figure out what I'm looking at. And then Stanford had this illustration, and I got this out of NCBI, and I think this is exon 2, 3, and 4. And
so that's the ones on the other side.
What's that? Yeah.
Those are the ones on the other side of the fusion.
Yeah. And I'm trying to figure out why. Oh, no, here they are right here. Okay. So this is. Oh, wait a minute. I don't know what I'm looking at exactly. But I was trying to find these, the H3K4ME3, which is sort of a transcription signal. And that's right there because. Oh no, this isn't mine. This is Tompkins, okay? So Tompkins here, okay, out of this, okay, that's that H3
fusion site, yeah, okay.
K4ME3. That's what he came up with. And then he came up with, these are transcription finders. Here's exon 1. Here's the fusion site. Here's another transcription factor. And so I went down and I pulled the real data, the current data, and I came up with a kind of a different picture, which disturbed me. Because here's exon 1. Here's your fusion site right here.
Yeah.
It's in here. The transcription starts right here. Kind of on the tail end of that.
Not in the fusion site.
Right. It doesn't seem to be exactly where he put it in his drawing. And he got exon 2, 3, and 4. So, yeah, and it just did not look right to me. And I know, he's got that much right. He's got this little piece right. But what did he do? And it kind of looked like he almost photoshopped it. But I would never accuse him of that. I don't know. Something's wrong.
Now, what's the
saying?
Never attribute to malice what could be explained by incompetence.
Right. I think it's incompetent. No, actually, okay, here's exon 1. Well, wait a minute. There's only 3 exons in this one. And he did this maybe a few years before I did it, and they may have a better picture, but I'm not sure. And then let's see, here's transcription, and I don't know. I pulled a whole bunch of other stuff, but anyway, that may be boring, but anyways,
not at all.
But here, when people argue about chromosome 2, this is the actual DDX11. Look at how many exons.
I mean, chromosome 2 is so frustrating to me because, you could try to argue about it, but it's not something that's worth disputing. And if a creationist is going to make disputing that a centerpiece of their argument, there's nothing I'm going to say that's going to convince them otherwise. There's nothing. There's no point there. Where there's other stuff I think there is more of an opening, but for chromosome 2, that's a slam dunk. Either you buy it or you don't.
There's more to this doc. I do have a bit of a slam dunk on the bottom that we should always bring them back to. But this is the fusion site, and I took the TTA GGs as much as I could and illustrated them. And I had to dig for that site. And these are the coding parts. But the thing is, I did a whole bunch of stuff. I spent way too much time on chromosome. Here's the only thing you need to know. This is all you need to tell a creationist. When he says that chromosome 2 is not a fusion, this is 2A, this is 2B on the chimp, this is the human. Look at the lines.
Yeah.
That's it. This is the rat. Okay. This is the entire chromosome. Actually, okay, that's the entire chromosome right there. all the way down, it matches. So either God smushed them together or something natural smushed them together. Either way, take your pick. But it's a fusion, nevertheless. God fused it, or
Yeah, absolutely.
how can they deny? And they never talk about the Sydney diagram. The only person I've ever heard bring it up in a debate is CRISPR. I don't know if you're familiar with CRISPR.
Maybe not. So my introduction to this whole YouTube evolution creationism thing was the debate with Sal. Like, I literally did not even know this community existed until like 3 weeks ago.
Yes, this whole
there's a whole. So I'm still learning my way around all the people involved. So I've maybe watched something that CRISPR was in. I don't know. I'd have to go back and see. I'm sure I will at some point. But
CRISPR was in some very early stuff with Standing for Truth, and he smashed him. He just crushed him because he's also a geneticist. He works out somewhere out east, and I've talked to him. I don't really know anything about who he is other than that he's CRISPR. So, but anyway, I mean, in a debate, this is all you need to show, and all this other garbage is just afterthought.
Need to show. Who knows?
Yeah. Yep. It's pretty nuts. Anyway.
So, what else do we want to talk about? We can talk about, let's see, we got genetic entropy. We got, I don't know if you've, let's see, we got Jensen, I still don't know how to pronounce his name, and mitochondrial Eve. We've got,
I think it's Jensen.
Whatever you want to talk about.
Okay. Well, it's a lot of things that I would all love to talk about.
Fitness, if you want to do fitness. And we got...
Something that drives me crazy is John Maddox has been bringing up this microRNA has shown a complete reversal of phylogeny.
That's
and
BS,
and then he won't link the paper.
That saga. I have heard of that saga.
Yeah, well, yeah, we're having a big fight over this, and so he won't even talk to me anymore.
Oh, really?
Nope.
I'm hoping to have a conversation with him at some point because he says a lot of stuff that's wrong. I think he's actually having a debate right now.
He is.
So and I'm sure there's a lot of misinformation going on in there because I've watched a few of his things and it's all the same talking points. And if you happen to watch this, that's logical, plausible, probable? Is that logical, probable?
Yep.
Whatever it is. Again, still learning everyone's names. LPP.
Yeah.
Come on and chat with me because like there's so much you say and it hurts. Right.
Yeah, I had a debate.
Sorry, I got a whining dog here. Go. There we go. There we go. All right. Sorry, the dog was whining.
And they screwed with me on Standing for Truth channel until I ended up doing it later than I wanted to, because I have about, like right now, my brain is running at about 30% of what it does at 4:30 in the morning. I don't know if you know, you get emails from me pretty early. And I had this debate with him, and we never got past. I asked him for a paper, and he got mad at me, and you know he doesn't like that. Even though he has provided, I have downloaded a lot of his papers. But anyway, so we came to this point where he brought up Dennis Noble's book, and it was the funniest thing because I had Dennis Noble's book open.
Which book?
Dennis Noble, you familiar with? I think he wrote it in 2017. I would have trouble finding.
Maybe.
Let's see, Dennis Noble.
All right, don't worry about it. I can look it up. Hang on, let's find out.
Okay. The latest one. It really isn't a bad book, but he had a quote from him.
Dance to the Tune of Life? Yes,
that one.
There
Yeah.
you go. Yeah.
Yeah, and Dennis Noble turns out to be kind of a character. He's the one that allowed that alien thing in with the alien jellyfish, I guess.
Oh, that every time that comes up, that annoys me so much because it's you can only make that art. I get very annoyed when people don't do phylogenetics correctly.
Right.
It's like, it's not alien. It's just different. It's recombined. It's something. It's not alien. And also, you can't take one little piece of the genome and be like, look, it's weird, and therefore it's a different thing. It's like, no, there's the rest of the genome that's nested. Like it's just, you have to when you're doing phylogenetics, you have to remove recombinants. If you don't remove recombinants, weird stuff happens.
Yeah.
Like,
PZ Myers on Dennis. No, let's see. On God, what is it? Anyway, PZ Myers did a video last week about that and about Dennis Noble, who he does not like at all. A lot of biologists don't.
Can imagine.
But now, John Sanford, or not John Sanford, Maddox, John Maddox. His whole thing is biosemiotics and information, and he apparently got a hold of a whole bunch of papers from the Royal Society, and he read them. And then he also got a hold of a paper by a guy named D'Onofrio and Abel. There's two of them, D'Onofrio and Abel. And they have, D'Onofrio has published over 50 papers. He wrote 2 books, which I own. And it turns out he works the director of some institute for some kind of research or whatever. Well, PZ Myers looked up his address for the institute, and it's some dude's house. In a small, and he's
not actually a thing, yeah.
Right. He's he has like several roles in these organizations, and they all are in the same address. Well, anyway, he's a brilliant man, and he wrote some really good stuff. He's the one that came up with ribosomal pausing. Him and Abel did a paper on ribosomal pausing, and he makes a case for pragmatics, for syntax, semantics, and pragmatics. And it's under D'Onofrio and Abel, and I have the papers. And so that's the beginning. That's kind of the first part of the thread. The rest of the thread is this massive amount of work by the Royal Society, which I don't know if you want to see what it looks like. It looks like this.
Yeah,
that's what it looks like. So I mean, clunk. It weighs about 15 pounds. It cost me a tree in a bush to print. But so all the stuff from Royal Society is on biosemiotics, and something that's been happening since probably about 2010 with this push into a different informational way of looking at biology, which is all wonderful and all apt, I think. Are you familiar with Dennis Noble, and are you a hater? Or?
Not, I don't have an opinion. There's things that I kind of glance at, and it kind of drifts by me, and then I say, you know what, I'm not going to form an opinion on that one. I'll let everyone else can fight over it. So there's some people that are like highly controversial, like Dan Grauer. Everyone's got an opinion on Dan Grauer. On that one, I'm in. On that one, I'm in. But like some other ones, I'm just like, you know what? No.
Oh, are you mad at Grauer too?
Oh no, I think Grauer's great. I agree with Grauer on most things.
Okay. Yeah, I'm looking for.
I in particular really like how Grauer gets kind of annoyed at other biologists when they're doing shoddy work, and the subtext seems to be like, stop making us look bad, which I appreciate.
Well, PZ Myers does that too.
Yeah.
He gets extremely angry, and he's really angry at just about everybody. But do you know PZ? Are you familiar with his blog?
I'm very familiar. I've been reading PZ's blog since the good old days of the Dover trial in 2005.
Oh, excellent. Okay.
Yeah. Back in the old science blog days.
Yeah. God, I don't know if I told the story on my channel. I'm afraid I didn't. It's going to be a repeat. But in 2008, I was working for JP Morgan out in Columbus, Ohio. And Ben Stein happened to be early premiering his movie at the Mall of America, 5 miles from me. And my son was sitting at home, and he heard that PZ Myers and Richard Dawkins were at the Mall of America, and they were going to the movie.
They were going to see the movie.
And Dawkins got in, but PZ Myers got tossed out.
I remember that story that they stopped him. Yeah.
Have you heard about this?
I remember reading about that story that they didn't let PZ in, but they let Dawkins in, which didn't make any sense. But yeah.
So okay, PZ Myers was tweeting about it. My son was reading the tweets at the same time that PZ Myers was tweeting. He called me and told me about this and sent me a link. I signed on to Richard Dawkins' forum. Well, Richard Dawkins was still watching the movie. And I became a member, and I did 11,000 posts there. I learned neuroscience and consciousness, and then I switched over to Rat Skep, where I did an additional 32,000 posts, and another place where I did. So I got like 50, 60,000 posts.
Well, since you mentioned Dawkins, what do you think of and this is one of my favorite controversies. What do you think of Selfish Gene?
I have never read it, but I happen to
highly recommended. Very good. Whether you end up agreeing with it or not, highly recommended read.
I have 2 copies of Extended Phenotype.
Oh, there you go.
One is a lot fatter, but this one is really tiny print, so I'm not sure.
Might be the same. Extended Phenotype's dry.
Do you recommend it?
The Extended Phenotype is drier. The Selfish Gene is really nice. It's he paints a really nice picture. I think it's really useful for explaining things personally, because I'm more on the microbial side of things. So you can look at whether it's a bacterial biofilm or even like cancer in the human body. I think Selfish Gene kind of paradigm does a nice job explaining stuff like that is otherwise tricky to explain. So yeah. I like that. But I know a lot of people do not. And I love arguing about that. It's a fun one.
Okay. I'm trying to get through a paper here, but I'm probably not going to find it right away. Anyway, let's see, primate chromosome and evolution. I'm in the wrong layer. I must have them somewhere else. I don't know what I did with them. Anyway, I'm not going to find it. But yeah, I'll check it. I usually don't read pop, but is that one deeper than just pop?
It's not super technical, but it does get into like the math of like an evolutionary stable strategies a bit.
Right.
And it's there are places where he'll say like, look, we're going to do this by analogy, we're going to do this in plain English, but here's what we're really talking about. And it's nice to like, okay, we're going to step back, we're going to illustrate it, and then we're going to jump back into the nuts and bolts of like actual genes. I think it strikes a nice mix and tone, and like I don't find it overly simplistic, or I should say I didn't find it overly simplistic when I read it. So it's I think a nice mix.
Okay.
It's good. I like it a lot. I think it's really good.
I'll take a look at it.
Yeah. And for one person watching, I think it's one person watching. Highly recommended. Selfish Gene, Richard Dawkins.
Okay. I wonder who it is. There's always that one person watching. I'm not sure who it is. Yeah, I don't have a very big channel, but I ended up picking up Survival of the Sickest. You remember Sal Cordova's talked about that?
I saw that in Sal's talk, but I have not read it.
I think his point is that it's complicated. It's complicated.
Surprise.
Life is really interconnected. We're interconnected with all these diseases and viruses, and they have surprising things, like sickle cell anemia, where something will seem like a really bad thing, but it'll end up allowing you to survive or allowing a race to survive. But he doesn't really make a case in the way that Sal thinks he does. He's not talking about what I think he is, but I just read a few pages in the beginning. I'm going to try to get to it, but that's also a pop book. So, But it starts out with an interesting story about his father dying of Alzheimer's and how he got into genetics because of it. So, medicine, I guess, and whatever. But
that's actually really interesting. Just talked about that in class just last week. That's a really interesting question of aging, and is it coincidental that we break down as we get older, or is it antagonistic pleiotropy, where you have things that are beneficial early on and then catch up to you later?
Right.
And it's one of those great questions where we don't have a good answer right now, and either way is perfectly plausible, and we just don't know. And it would be really cool to find out, because if we want to try to extend the human lifespan without doing things like selective breeding, that are horrifically unethical, that you can't do, we're going to need to know what the answer to that question is. And right now we just don't know. And it's fascinating.
Yeah, I'm hoping that if I start losing my mind, it is going to be tragic and it's going to be spectacular, because it's probably not all going to go at once. It's going to go in pieces. And they're going to be pieces that pop out, you know, so it'll be pretty amazing. But I tend to be less and less correct the older I get, and I'm getting to the point where I really don't give a damn what I say online or to anybody or
who knows. Politically correct.
I'm less correct, less any kind of correct. So you never know what's going to. Well, my clients, I've been a computer consultant for 40 years, and I've had about 5 or 6 clients say, you know, that guy, you never know what's going to come out of his mouth. Because I'll say things to my clients that shock them. Particularly when I worked in South Carolina or North Carolina for Wachovia. Mm-hmm. I worked in Charlotte, North Carolina, and I was sitting at a desk talking to somebody, and I mentioned atheism, and she says, "Oh, be quiet, be quiet. They'll hear you." And of course, being who I am, I didn't stay quiet. But they couldn't fire me because I was too damn good at what I was doing. So, I don't know. But I couldn't help myself. I just can't help but, you know, somebody says, "Don't touch that." I gotta touch it.
So I just today was informed of an interesting exchange. I don't know if you saw this with Jensen, and there was a publication. I don't even know when it was. Recently, I guess it was in education literature about how this idea of like hyper rapid speciation following the flood. Have you read any of that?
No, I haven't.
So this is the idea that you get following the flood. You get, well, you could only have so many things on the ark, and you look at things like dogs and wolves, for example, and it's like very obviously the same kind, and so like they're going to share a common ancestor at some point in the past. And what we're getting to is the authors of this piece, they introduced this idea, I think they called it post-creationism. It's this idea that you're getting down to so few created kinds that you end up having within the span of a few thousand years, hyper-rapid evolution that involves speciation literally every generation to get your current biodiversity. And we're getting to the point where there's so many, there's nowhere to draw a line. If you do the kinds, there's nowhere to draw a line. And I'm sure you probably heard the phylogeny challenge of like, where are the actual breakpoints? Right. And what we've been seeing is that those breakpoints just move further back in time as we go. Yeah. So it's like, okay, it's, well, dogs and wolves, but not this other thing. And then, no, you include all of them. And then it's like, okay, all the felines are going to be together. And then pretty soon it's all the carnivores are going to, and it just goes and goes and goes. Yeah. And you end up with, you know, this, you only have 4,000 years to get thousands of species. Well, how does that work?
Well, maybe you can check me on this, but I had a hangout with Standing for Truth on this Why Did God Make YECs series. Mm-hmm. And we were talking about created heterozygosity.
Oh, he is so wrong about that. It hurts every time I listen to it.
You can only have 4 alleles at the maximum at any one locus.
Or maybe 14, depending on the species.
Why do you need 4 different insulin alleles, right? For a particular gene, so a lot of the genes wouldn't matter.
Yeah, right.
Heterozygosity. But then he was telling me, oh, no, you have thousands of other genes that could have, and it's like, no, nope. Not at that locus.
Nope.
You know, maybe an occasional gene conversion, you might have something happen, but so you've got a bottleneck. And then he calls it, and then he said, yeah, but we had an extended bottleneck, and he didn't after the arc. It was explosive growth. But somehow when we had the bottleneck crossing over, you know, into Europe, that was extended. And I'm asking, why was it extended? You mean they didn't have sex for 20 years? You know, they had less sex when they crossed over because they were cold, or they were in Europe, and maybe because they were cold, you know, they didn't want to mate as much. But he never could tell me what this extended bottleneck is. And then he keeps saying that, well, you got a real problem. You got this bottleneck of 2 to 10,000 people, and it was probably more like 10,000 to 100,000 that crossed over, or 10 or 20 or whatever, in small groups. But that is not a that is not a bad, you know.
It's not, in terms of the effective population size, it's fine. In a bottleneck, what matters is the effective population size. It's not the raw number of people. It's how extensive the gene pool is and how many people you need to maintain that gene pool is what matters. The breeding population is what matters.
Yeah.
So it's just going on raw numbers is not the relevant metric there. And if you happen to see this, let's talk about heterozygosity, because like, for real, you got to get this straight. I've heard you say it like 3 or 4 times, and like it's wrong every time. It just never makes sense.
On his hangout the other night, he was answering a question that Erica asked him, and the question, Erica said that cats and tigers are have more differences in their genome than humans and chimpanzees.
Sure.
And he said, okay, we can explain that. So here's how he explained it. When God made animals, he didn't just make 2 like Adam and Eve. He made thousands and thousands of them, right? And they had all this created heterozygosity. And that's why house cats and tigers are so widely separated. But I wanted to reach into the screen and grab him by the throat and say, what about the ark? You only had 2 kitties on the ark, right?
Yep.
Where the hell did your heterozygosity go?
How much heterozygosity you start with if you go through a bottleneck of n = 2.
Yeah.
Doesn't matter what was on before that.
And no matter how much heterozygosity is stuffed in there, it's not going to have the effect that you think is. So I really suspect he doesn't understand how genes work exactly. I think he thinks they shuffle around or something. I don't know.
Some of them do. Some of them do. Most of them don't.
I was trying to get him down to the point where he would look at it, but then he wanted to move on to another subject.
That is a common strategy.
Common strategy, right?
Very common strategy. It's his go-to is the whole is that goo to you? Is that what is it? Bacteria to biologist? Is it fish to fisherman? He's got a million of them.
Right.
And it's always okay, I don't have a thing, but let's move on to the next thing.
Yeah, they start talking about biogenesis, and then they want to talk about bacteria or fish to fishermen, and it's just absolutely crazy. Another strategy that he has, he does it, and we talked about this. He does the Gish Gallop, and then the last thing he throws out is the bait. It's the red herring, the chum, so that you'll go for that,
Yeah, you gotta. It's pinning people down and staying on task is like the thing in these conversations. Most things are just blind alleys that don't matter. If you can, but you got to pick your topic and narrow topic. And I'm like, I've told people, like, I don't do like this kind of like the thing with Sal when we're talking about genetic entropy. Like, I'm not going to do like evolution versus creationism. We could talk about genetic entropy. We could talk about coalescence analysis. We could talk about, whatever. But we're not going to talk about evolution and creationism because that's too broad. We're going to, we can talk about macro evolution, which don't get me started on that. Or if we can talk about it if you want. But like, we're going to pick a specific topic and we're going to drill down on that. We're not going to flip from thing to thing every 10 minutes because you don't accomplish anything then.
That's what I like to do, is drill deep into just deeper and deeper into one thing. And what I find with them is that if you skim below the surface and you hit that point where their knowledge runs out, where they're basically their answers in Genesis runs out, which is kind of where they get everything. Then you end up, they just toss you off, and they'll try to bait you off. And if they can't get you off the subject, Standing for Truth will say, well, we don't want to go down that rabbit hole. Or
yep. It's like, no. Let's go down the rabbit hole. I insist.
Let's go down a rabbit hole.
Let's do it. Let's take 20 minutes and really get down on this.
Yeah.
Oh, 100%. Yeah. And you see that with everything, whether it's, define information. I literally had someone the other day, I asked what the definition of information was, and the response was, we all know what we're talking about.
Right.
No, I don't. How do you put a number to it? How do you quantify it? I don't know what you mean. And like, we can't talk about the rate at which it accumulates, or the rate at which it is lost, or the maximum amount that can form if we can't quantify it. Like, don't just move on to the next thing. Give me a number and a technique. Give me a metric.
So one of the papers that Cell turned me on to was by Didier Aubiff. It's Aubiff, or no, Aubeouf. It's I believe it's in that folder I sent, and the paper is thick. He has an amazing amount of illustrations, and I think that was the one where he was going into. He went into, I think, deeply. I'm pretty sure that's the one. He went deeply into how the tRNA and the protein and the DNA, I think, all connected, and they sort of well, you had all these different ways where the chemistry decided what the codons were going to be.
Mm-hmm.
So the codes, like the big thing about the informational people, the Royal Society people, and biosemiotics, is that the tRNA makes a break with the DNA, basically the RNA and the proteins. So there's a code translation going on. So it's a purely informational thing, right? And they call
it's just based on affinity.
Yeah, but no, this Ubif was making the point that is not random at all. That is really preordained, and he was going through all these different sequences. And then the other thing, I think it was like GC content will create alpha helices, and AT will create beta sheets. More often, these richer areas, and what we find is that there are statistically areas that are statistically off center. They're not completely neutral.
Mm-hmm.
And he was making the case about how proteins started to get created, how these structures were created, and it was a very interesting paper. But you take a look at that one; that was interesting.
Yeah, and absolutely look it up. Thank you.
I think it's in that bundle I sent you. I think it was one of the first papers I picked out, and it's very, very thick, and it's quite a read, but that's very good. And there was something else I was going to get to after that. This thing about created heterozygosity, I'd love to model it, and I can do that in Python, but it's a very difficult model, to model a tribe of humans that are mating. And I started working on it one time, and I think I'm going to switch over and do it in Ruby, which is a better language.
Uh-huh.
And I'm going to try to create a model of Adam and Eve and just see where it goes,
So simplify things. You assume random mating. You start with Adam and Eve heterozygotic at every locus.
Every locus, right.
And then so you get 4 alleles total for every locus across the entire genome. And then random mating, and just go from there and see how long it lasts.
Yeah, I think I started to model that, and then I ran into problems because I couldn't stand the idea of incest. So I started looking at my network.
So then you have to impose restrictions on the mating.
Yeah, and it has to be a kind of restriction where you go down the node, like this node to that node to that node. You got to have a distance of 4 nodes. It's okay to lust after your second cousin, but not your first cousin. Right. So anyway, I got into that, and then I got busy on something else, and of course my mind wandered. So, but I will get back to that. But I wanted to do the whole model. We got 4,400 years, and I had a mating system set up, where there was they would start mating at around anywhere randomly between 16 and 22, and keep mating until they're about maybe 28, 30. And then, of course, they have the thing where people used to live like 900 years or something.
Yeah. So once you add that into it, it gets all messed up. And I would omit that from the model. I figure that's probably not what's actually happening. Although, there are lots of creationist geneticists that include that. Stanford explicitly includes that in genetic entropy as an actual thing, where he has that graph of the lifespans dropping off. And I think that actually is a good illustration of part of the problem here, which is that these arguments that we're responding to as scientific arguments, and I said this when I was talking to Sal, that we're going to treat this as a scientific hypothesis. And even though Stanford kind of winks and nods in that direction, that book is not intended for a scientific audience. And when he talks to a scientific audience, he knows that what he says in the book is ridiculous and will be laughed at.
Yeah.
It's not intended to be taken seriously. And that goes for a lot of people. That goes for Purdom. That goes for Jensen. In the whole thing with the mitochondrial Eve time to most recent common ancestor that Jensen does, that is not intended for a scientific audience. Because like reading what he says right in that original paper from 2015 that like, I don't know, maybe, but we're not really sure. He says it right there in black and white, but you need to know what to look for in what he's saying. And to a non-scientific audience, what he's saying, it doesn't come off as a disclaimer, but to a scientific audience, he right there in the discussion has a disclaimer.
Have you read that book?
I have not read the new one, Replacing Darwin. No, I have not read Replacing Darwin.
You haven't read my book? My book. Did you see the Herman Mayes debate?
No.
Oh my God, it was hilarious. That's all Jensen said was, "It's in my book. Did you read my book? My book. Did you read my book?" It was like,
for me, please. It's like, come on, man.
It was on Modern Day Debate. It was pretty good. Herman Mayes.
Oh, Jensen went on Modern Day Debate. Maybe I can get him then.
Herman Mayes.
Be a real short debate, though.
Be short, yeah.
Well, the only thing is you used a mutation rate instead of a substitution rate to do coalescence analysis.
Yeah.
Yes or no? Like, we can go through the technique, and it's like, no, he used a mutation rate, not a substitution rate. And like,
that's what Herman Mayes said.
That's it. That's the whole thing. He used the wrong number.
Yeah.
And I say wrong like it was a mistake. He used the wrong number on purpose to get the answer he wanted. It's not like he actually knows how this stuff works. And we know he knows because he said straight up, I'll pull the quote. He said straight up,
wasn't he using D-loop pedigree studies?
He was also using D-loop pedigrees, which makes no sense.
Right.
You can't use the D-loop. But he said, I asked myself, how can I use and abuse my training to influence eternity rather than for temporary gain? Like, he just straight up said, like, I'm going to lie about it. Like, I have a PhD, but I'm just going to lie. He just said it right there. That was in his welcome little bio at ICR, at the Institute for Creation Research. That was like his, like, welcome aboard, Dr. Jensen. And like he just said straight up, like, I'm going to lie to all of you.
Yeah.
Like, nobody bats an eye.
He's probably pulling down 150K a year doing that.
Man, should sell out.
I would lie too.
Should sell out. I'd make a lot more money if I sold out.
Yeah. If I were a good liar, I would become a creationist and I'd go to work for them because I know enough.
I swear, I could make the arguments as well as he does.
Yeah.
I could probably do it better than him.
Yeah. Amazing. But there's that D-loop thing, and then there's the Parsons paper, and I in the FBI and the Parsons paper, and I think the Parsons paper wasn't that about some digging up graves of Russian czars or something? Was that the one?
What was the context for that? Was that still Jensen with like the migration patterns? Was that thing that
Standing for Truth?
Standing for Truth.
His basic argument. It was Deadly Dakota Raptor. Oh, hello, Deadly Dakota Raptor. Subscribe to my channel, and I'm not sure who Deadly Dakota Raptor is. I think I did subscribe to Deadly's channel too. Anyway, welcome.
Deadly's one of the main minds over on the debate evolution subreddit.
Really?
Oh yeah.
Okay.
Come hang out on Reddit sometime. We have fun.
Yeah, Reddit kind of. I don't know. It just. Well, for one thing, I don't like the way it's formatted, and it's too hard to keep track of arguments and the descendants and of all that. So anyway, friend of Dan.
Right.
Cool. So nice. Deadly Dakota Raptor. I should. Which Reddit are you on?
It's a debate evolution. And while you're looking it up, let me pull up. I'm working on. So my next thing is going to be on Jensen and mitochondrial DNA because. Standing for Truth just did a thing on this, and I told him, "Look, you're way off base on this, so I'm going to do a thing on." It's a work in progress, but let me see if I have this open here. I can just pull this
have a bundle of papers on that.
Here's the. Oh no, that's not the right one.
Including persons.
Here we go. Here's the problem right here. Let me just pull this up. I'm going to try to not break the screen share. Yeah, I know how to do this. Let's do that. That's the one. Yeah, let's look at that. So if you look at this is what we want to do. When we're doing coalescence analysis, like to calculate how long in the past the mitochondrial most recent common ancestor was, you use substitutions. You use substitutions that are transmitted via the germline. What Jensen does is he uses a pedigree study, and he just tabulates all the differences between the parent and the offspring. So you catch a bunch of somatic mutations that are not going to end up persisting in the lineage.
That's what
And
I
like,
suspected, but nobody
that's
ever
it.
talks about that.
Yeah, that's it. He uses, it's that Ding 2015 paper. It's a pedigree study. And all he does is just tally up. Here's the things in the parents that the kids don't have. Here are the things in the kids that the parents don't have. And
Well,
he just tallies all those numbers up and says, okay, that's the one generation mutation rate. No, most of those are somatic mutations that are not in the germline.
yeah,
That's not how you do that. And he
yeah.
knows that because he
I
says so
suspect
in that paper.
I suspected that was the case, but nobody ever talked about it. And it's like, how in the heck do you do a pedigree study and not account for somatic mutations? That was
He
amazing.
he 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 says that that's a problem. He acknowledges it. Let's see. It's Jensen, 2015 mitochondrial whatever recent. Let's see.
Is it one of the Answers in Genesis things?
Yeah, it's one of his AIG papers. Here we go.
Published.
Let's see if I let's go down to the bottom because it's literally the last thing in the paper. It's like right at the end of the discussion, and it's just like it's a disclaimer. It's a by the way, we're not sure.
What version?
No, that's not the one, but there's another one.
He wants to know what version of PowerPoint something is.
Oh, I have got an old version of everything. I'm on, I don't know. I think I'm using, I think it was 2011. I think it's still 2011 version. Yeah.
Okay. So I wonder. Okay. So that makes a lot of sense.
One.
Like when I put it out a whole bunch of papers on mitochondrial DNA, and it was kind of a mess as to trying to figure out what the rates were. And then a part of me was saying, why? What about nuclear DNA, which is a lot more sensible in a lot of ways.
Yeah, I mean people like mitochondria because those are recombined, so it makes it easier. But
yeah, it makes it easier for coalescence studies,
right? Because you don't have to worry about dealing with recombinants, which mess with your analysis.
You should do a really good educational video on coalescence. I've got a book called Coalescence Theory, and it looks impenetrable. I'm afraid of it.
It's really intuitive. It takes time to wrap your head around. Like the idea of like, here, I'll show you it. Let me pull up PowerPoint again. I'll give you a different slide this time though. Just to show you because I gotta, obviously if I'm gonna do this, I gotta like explain it. So like, we gotta look at like the idea of a most recent common ancestor and what this figure shows. And I don't know if this is the final form or not, but the black represents the most recent common ancestor for the living generation, which is down here, right? So, not the first member of the species, not the only individual alive at the time. There's lots of other people alive, not constant in time. This coalescent point moves. So if you go back to this generation or this generation, you get a different most recent common ancestor.
And every gene is going to have a different coalescent point.
And I love that you said that because that is literally the next slide. Let me go back to it because that is literally the next slide is, by the way, when we're talking about mitochondrial Eve, that's only for the mitochondria. The rest of the genome is different. So you can take Jensen's numbers at face value and do the same techniques for other parts of the genome. And you get answers that destroy the young Earth chronology anyway.
Right.
So like it just.
Yeah, but the coalescent theory, there are ways to measure the distance between two different. That's the part that I don't know much about is how to use coalescent theory.
When you say distance
between nodes,
you mean like time, genetic distance?
Well, for phylogenies,
it depends. It depends what you're going for. So you could measure. Whenever you have like when you do a phylogeny, you always have there's always an unseen axis that's either going to be time or it's going to be genetic distance. Number of substitutions or whatever. And depending on your data set, you may or may not be able to assign time.
Yeah.
So like the nice thing with the mitochondrial data set is we can correlate specific mitochondrial alleles with specific migration events. So like, okay, we know humans first settled this island at this time.
Okay, my cats are making weird noises and I have raccoons that invade my house every now and then. They invade my cat fence. And I just have to keep an eye on them. It's raccoon.
I only have a couple more minutes, so if we want to wrap this up, we can do that.
Yeah, we can do that. But I would like to get to know Deadly Dakota Raptor. You should email me if you're still watching at speedofsoundofgravity@gmail.com, and we'll have a chat. But anyway, yeah, there's a lot to cover. There's so much to do, and I guess I would like to get better at content and take on each of these little creation points. And it would be, you wonder if it's a waste of time, but then what I know people who have become creationists recently, and they have children, and it scares me.
Mhm.
And I think, well, if children are being taught this garbage, then we need to do something about it. And it takes 10 of us for one creationist, right?
Yeah, it really does. And that's my approach is this very, very, like, obviously I'm not aiming the slides I just showed you at like a technical genetics audience. And I have no illusions that if Jensen is ever going to see those, like, it's going to change his mind. But what I want are the people that are, somewhat interested, not
Borderline.
experts,
Yeah,
but
the
like,
borderline.
do it for the lurkers. You do it for the lurkers. And that's where the value is.
Yeah, the thing is, I decided, okay, Felsenstein used the word evolutionist. So and they're always doing this. You're an evolutionist, and it's your religion. You have a belief. So I decided today that I am an evolutionist. It's my religion. I have faith. It's my belief. Let's go from there, and we'll see what happens. But
that's an excellent strategy. Also, is just concede the point. It's like, well, don't make me define kinds because you can't define species. Okay, great. I can't define species. Species concepts are stupid. Move on.
The interesting thing is, I got into one big problem I had with evolution and biology in general is how genes construct organisms. So I spent a year and a half on P.Z. Meyer's embryology books, Morphogenesis, and I learned how embryos are created. And once you realize that, the reason I believe in evolution is not because of fossils or evidences. I got to do my it lost me again. I don't know what the heck is wrong with this thing. I don't know. We'll see if it gets me. I'll be fuzzy. I am fuzzy. But anyway, the reason that I believe in evolution is not because of some random package of evidences, as they call evidences plural. Does anybody else use that besides creationists? I don't know. Anyway, that's not why. The reason I believe in it is because I understand chemistry. I understand molecular biology. I understand embryology. I understand how it's possible, and I understand how it happened. And abiogenesis is the same thing. The more I learn about this massive collection of crazy molecules that you find in coal tar and in asphalt, and the more I look at heterocyclic organic chemistry, I can imagine very easily how life may possibly be inevitable when you have the right temperature range.
Yep.
And that's why I believe in evolution, not because of evidences.
Nice. I think that's a really nice place to end it, and I need to be going. So
okay, good. And if we could talk,
thank you for inviting me on.
If we could talk for just a minute afterwards, then I will end the broadcast now.
Absolutely. Yeah, totally.